Okay. Thank you. So we have to continue the session, and now we switch to the video aquatic session. And we have the presentation by Gilles.
Good morning. So I'm going to talk today about microalgae and photosynthesis for microalgae. And we have been, since for a few years, we have been working on the CO2 concentrating mechanism of microalgae, and I'm trying to explain you what's the interest for the climate. So, as you know, part of the CO2 capture by terrestrial ecosystem are done in aquatic ecosystem on the microalgae, and which, when we call microalgae, we include microalgae and cyanobacteria. They can take up to 50% of the carbon CO2, which is fixed by ecosystem annually. And what's specific to aquatic ecosystem is that CO2 can be very limiting for photosynthesis, because for different reasons. First, because of the specific factor of Robisco and the low solubility of CO2 in water. So, which makes during the time course of evolution, CO2 concentration in the atmosphere dropped, and algae had to develop a mechanism to concentrate CO2 to make Robisco functioning at a maximum level. And so one of the reasons is the low solubility of carbon dioxide. The other is the low diffusivity of CO2 in water. So, which means, and that's the next slide. So during the time course of evolution, CO2 concentration dropped, and different algae and cyanobacteria, they develop independently CO2 concentrating mechanisms to make Robisco functioning at a high level of CO2 fixation. And this CO2 concentrating mechanism, although they are different depending on the species, they share common properties, which are bicarbonate transporters, carbonic anhydrases, which catalyze the exchange between CO2 and bicarbonate, and most of the time, a compartment, which is called pyrenoid or carboxysome in cyanobacteria, where Robisco is packed in a fast separation environment, which allows to concentrate CO2. So, and this is most of the time an inducible system. When you grow algae under high CO2 concentration, the affinity of photosynthesis for CO2 is very low. But if you grow them in low CO2 environment, in air, you do see that you have an increase in the affinity, so which shows the induction of the CCM, the efficiency. And it has been early recognized after the discovery of CCM in the 80s that it needs the energy of photosynthesis to concentrate CO2, because we need energy to concentrate against the thermodynamic to make a gradient. So, and the question we asked a few years ago was, how is the energy of photosynthesis is transmitted to this CCM? So, briefly, you have photosynthesis here from PS2 to PS1, and what generally the most common electron transfer pathway is from water photolysis to reduction of NADP, and together with the proton gradient. Proton gradient is used to make ATP, and the reducing power and ATP are used to fix the CO2 through the carbon metabolism reactions. And additionally, and we've been working a lot of this, we have additional pathways, which are cyclic or pseudocyclic electron pathway, or interaction with mitochondria, which allow to generate additional proton gradient to make more ATP, because it has been recognized that photosynthesis needs more ATP than just produced by the linear electron flow. So now, if you look at the CCM, so that's photosynthesis, and the CCM are two different systems have been in the model algae we're working on, which is chlamydomonas, which involves bicarbonic carbonic anhydrase and bicarbonate transporters. And by using mutants affected in the different pathways and measuring CO2 exchange, we have been able to show that part of the proton gradient, which is generated by this additional system of photosynthesis, is used in the thylakoid lumen to convert the bicarbonate back into CO2, because CO2 is fixed by the rubisco. So it shows that part of the energy of photosynthesis is not used for CO2 metabolism, but for CO2 concentration. And this is part of the proton gradient. But also we were able to show that the mitochondria contribute to the supply of energy to the external transporters, most probably this plasma membrane transporter of bicarbonate, by allowing a transfer part of the reducing power generated by photosynthesis back to the mitochondria, which make ATP. So, and we were able to evaluate the contribution of the different pathway to the energy supply of photosynthesis to the CCM. So now, what's happening when many people believe that because algae have a CCM, photorespiration is not functioning in algae. So what is photorespiration? Because you have at the rubisco level, rubisco catalyze carboxylation of CO2, but also the oxygenation. And when it's carboxylation, you have a CO2 fixation metabolism. When you have oxygenation, you have photorespiratory metabolism. And most algae are not able to metabolize all of the glycolate, which is produced by photorespiration. And part of this glycolate is excreted out of the cell. And our working hypothesis and the idea we want to develop at the moment is that depending on the conditions of the species, because in the different species we have different CCM and different energy requirements, the activity of CCM may vary in the environment, and which may trigger photorespiration, which has been widely overlooked because most people consider that photorespiration in algae is very inefficient. And that's something we are exploring at the moment. And one paper was published a few years ago and showing working on the bacteria which are found in the ocean and which have developed a very efficient metabolism to catabolize glycolate. But still, in spite of the presence of bacteria which use glycolate as substrate, that's a summary of different studies which shows measurement of glycolate concentration in the ocean, showing that we have relatively high level of glycolate in the sea, and which increase when you have bloom, that algae develop a lot. So, which to me shows that photorespiration is active, and that CO2 fixation by ocean could have been overestimated. So what we are doing now in the lab is to measure photorespiration, and the way to measure oxygen uptake when you have photosynthesis, because photosynthesis producing oxygen by water photolysis. So we use isotope and 18 isotope on membrane index mass spectrometry to measure the flux of entering oxygen produced by photosynthesis. And recently we developed, I'm almost done, a collaboration with people from Genoscope, because now we have a huge data of, thanks to the Terra Ocean expedition, a huge data of metagenomic and metatranscriptomic. And one of these algae that I've been working on, Pelagomenas, has been found in most of the sites I've been sampling. So what we are doing at the moment is to try to perform at the same time research in the lab to measure this gas exchange in the species, and to evaluate the role of CCM in the functioning of photosynthesis and photoaspiration, and try to look in the different conditions where this algae has been formed in the natural environment to make a link between the expression of the genes of CCM and photorespiration and environmental conditions like CO2 concentration, temperature, light. And obviously, we miss data on the glycolate concentration in the ocean, and one of my aim in the future will be to have more information on this glycolate, particularly when you have a bloom of algae, which are things at the conditions where photorespiration is highly active. Thank you. And finally, thank you to my collaborators, Kadarash. Adrien was still was a PhD student in our lab and now working in Carnegie, and our collaborator, Jean Scope and Jake. Thank you for it.
Thank you. We have to move to the next talk. She's going to be given by Dan. Yes. We keep the question for the end of the session.
So good morning, everyone. My name is Daniel Chevrier. So thanks just for the organizers for having this today and yesterday. It was very nice to meet you all, and I'm happy to be able to present some of my work today. Thank you. And so I'm studying microbial biomineralization. I'm interested in a number of things, from the formation of biominerals to their biosignatures, how they differentiate from synthetic minerals of similar composition. And finally, the function of these biominerals, what are they used for, or what we could be using them for. Today, I will give something quite broad since we're limited for time. So I will present two different systems. Today, I'll present a little bit of magnetotactic bacteria. These guys are found in aquatic environments, also sediments and soils. They produce these magnetite nanoparticles, which are magnetic. And actually, so they use this to use essentially to create a little compass that they can use to navigate in the environment because they prefer oxic and anoxic conditions. We'll actually hear about them a little bit later. And what I'll talk more about is coccolithiform microalgae that make these beautiful calcitic plates of calcium carbonate. And besides these two models, I'm coming from a, And so I'm coming from a background of more chemistry and physics. So I apply often synchrotron-based imaging and spectroscopy methods to study the mineral composition, the formation and structure. And I'm more interested in nanoscanning and multimodal techniques because we are dealing with really nanoscale type materials. And I'm also interested in developing microfluidic setups to perform experiments in situ, for example, following mineral formation. And so, as I mentioned, these guys make a chain of magnetic nanoparticles, which creates kind of a little compass needle. But the other question that's interesting is that the coccolithophore, they also make somewhat of a cocosphere called, and I think that's more of a shield. The function of the shield is actually not 100% sure yet, which is an interesting topic. So one example of the studies with MTB, we are interested in looking at the composition of magnetite produced abiotically, so just in a chemical reactor, versus the magnetite produced by the MTB. And so we used a high-resolution ICP-MS to look at the trace metal composition that enters the magnetite crystal. So we measured the solution after formation and the crystal itself. So if we plot this with lattice strain calculations, we see for abiotic magnetite, so the dark circles, it fits quite well what's predicted. So if we have cations of different valence, 1, 2, 3, 4, we see often it does predict where they should be incorporated into magnetite. However, for the bacteria, which are these symbols with the open color, we see it's heavily reduced incorporation because it has to pass this membrane, it has to pass through into the cell, selectively uptaken. There's a number of things we did find from this study. Interestingly, there are a lot of incorporation of 3D metals for the abiotic case. But the other thing that we wanted to point out was that this approach we can kind of use to, if we want to modify the properties of the magnetic particles, we're really limited for what we can do physically. So we kind of have to be creative or take advantage of what we can do here. So I'll talk mostly about coccolithophore microalgae, because I think there might be some more connections here. They're really beautiful to look at, of course, but inside each of these shells, you do have a cell that is performing this biomineralization, where they form coccoliths one by one. And so beyond the kind of nanometric scale, these have an impact on our environment. So from previous biomineralization to today, we have coccolithophore blooms that we can see from space. So obviously their activity in our planet is of importance. And in particular, the importance comes from their ability to both photosynthesize, but also produce these calcific plates. And just to quickly summarize, essentially, these cells will be living in the photic zone, but they will die, and they will shed their coccoliths, which will sediment to the bottom of the ocean. When they do this, they actually create a pretty big sinking flux, dragging down a lot of organic debris. And this is something that a lot of climate and geoscientists are interested in, because it is a really important part of the biogenic carbon flux related to the blue carbon cycle. Obviously, I show something very general because this is not my area of expertise, but I'd like to show the connection between calcification and this kind of flux modeling. So I don't have to say that it is important to study these calcifying microorganisms in the oceans, because we are going through a kind of a climate change at the moment. So the interesting thing for us is what will be the impact on calcifying microorganisms, especially with the increase of CO2. We have a decrease of seawater pH, and this is going to change a bit the ocean chemistry. So how do these microorganisms handle such change? It's quite gradual, but there are other types of issues at the moment with ocean acidification and climate change, being decreased lower pH, warmer waters, higher illumination, and also nutrient stratification because of this warmer water regions that are formed. So the questions I have are how can, a lot of people have these questions, but how will these impact coccolith production, seeing as though they make such a huge quantity of oceanic carbonates? Obviously, this is very difficult to answer, but it's kind of a motivating aspect of why we should study calcification in general. And so what do we know about coccolithophores and how they calcify? A very general mechanism is that they're essentially recruiting calcium bicarbonate ions through different types of organelles to this coccolith vesicle, where they synthesize the calcium carbonate in a way that's different than any other organism. They actually use it in an ion by ion creation, which is really different from what we see for other microorganisms and organisms, which is more of an amorphous to crystalline transition. So already this is quite unique. And perhaps these bodies are really playing a role in controlling the growth, because it is a very, very ornamented, delicate looking structure. In the end, when the mineral is finished, it's then pushed to the outside and you create this cocosphere. So the last few years have been very exciting because there's a number of advancements with electron imaging that have shown different types of bodies that are recruiting calcium and potentially other molecules to help build these coccolith crystals. For example, there have been such things as coccolithosomes found near the forming crystal, dense calcium and phosphate bodies, which are also found when you are calcifying the coccoliths, and also more dilute phases, which are more stored in like a vacuole. So many different types of observations, but always just a single point. So the questions I have are, what are the roles of these bodies? Do they really play a role in controlling the formation? What are the effects of the chemical environment around the cell? Does it even matter? Are they able to really control all of this intracellularly? And what can we extract from in vivo? This information has been quite useful, but we really don't know the dynamics of how this process happens. So I will just go for a little bit because I think I'm sure we're close to the end. What I'm focusing on is techniques at the synchrotron that really provide the best of combined resolutions temporally, spatially, and of detection, so spectrum. And so I'm using X-ray fluorescence, even though it does not look as pretty compared to the beautiful electron tomography, or even for the confocal microscopy, which we all use. We're working with something that has the adequate spatial resolution to essentially resolve the coccoliths forming and the bodies next to it. And we can also measure things in minutes. Since the coccolith process happens over an hour, there's ways we can follow this at the single cell level to really follow the dynamics between the bodies forming and the coccolith forming, while also having elemental information like calcium and phosphorus and other metals. So this is performed at synchrotrons. Even here in Soleil, there's a beamline nanoscopium, which has this technique, which I've already used. But really the principle is to have a very small beam that scans your sample of just tens of nanometers. You collect elemental fluorescence like EDX, but you can also combine it with more other techniques like diffraction-based imaging. So here I again show a single cell of coccolithophore during calcification. And so what I'm working on now is combining this method by following the cells in situ. So I'm actually using optical and soon Raman with X-ray imaging to follow the formation of calcification under different conditions. So this is an ongoing project with ANR. And last, I'd just like to give a summary of the things I'd like to do in the future, just to kind of give a bit of, generate a bit more discussion later. Similar to the first study I showed with magnetite and the metal composition, I'm interested in doing a similar study for coccoliths to see how metals are uptaken into the crystal, what control do the algae have on preventing certain metals, and can we maybe use this for application after? And a few other examples are, I'm now kind of working more with the photosynthesis and trying to understand calcification and photosynthesis, how they are working together or separately, what the role of the coccolith might be for modifying the photosynthesis activity. And lastly, we're about to have a Raman microscope in our platform at the BIAM, and so there'll be more projects on that. So thanks very much and happy to discuss with you later.
Okay, we continue with Marion.
Okay, so good morning, everyone. My name is Marion Gehlen. I'm a PhD student from the LSCE. And during this PhD, I got this great opportunity to work with the BIAM laboratory and especially with Daniel Chevrier on a project about coccolithophore's biomineralization process and its relationship with photosynthesis activity of coccolithophores. But first, I'm just going to give a general approach of what I'm doing. So biocarbonates do form from what is present in their environment, so water and DIC. Isotopic will fractionate during calcification process, and this fractionation is generally linked to environmental parameter. So as a function of temperature, for example, for delta 18O, and this is the base of paleoclimatology. The fact is that metabolism, metabolic process such as respiration, photosynthesis, and enzymatic activity to also fractionate carbonate and oxygen. And this biological signature can be imprinted in the biocarbonates. So the resulting isotopic composition of the carbonate will be the sum of these two processes, climatic and metabolism, that we need to decouple in order to better reconstruct climate or access metabolic change in activity with climate change in the past. So this is what I'm typically doing in my PhD. So Dan already gives you a bit about introduction on coccolites. Just to give a bit more information. So they're a primary producer. They live in the first meter of the water column, and they appear during the Triassic. So they have an extensive fossil record, and they have this ability to capture CO2 from the atmosphere by photosynthesis. But what makes them very interesting is that they're able to produce a calcium carbonate plates, offering the opportunity for sustainable carbon storage. But before this solution can be applied, we need a better understanding of how coccolites do mineralize. and you will see the connection with Dan, and how environmental changes control their production. Something you may not be aware of is that coccolites, from a part of this bad student, we cannot use directly for paleoclimatic reconstruction, has very impacted by vital effects. And some researchers have shown that the delta frequency, for example, of coccolites of first place is largely influenced by biological activity, and in particular by photosynthesis. The magnitude of this disequilibrium, I'm gonna call it like that, is also a function of environmental parameters such as pH, atmospheric CO2 pressure, and temperature. The problem is that just from the delta frequency and the delta 18 record from coccolites, we cannot dissociate biological effect from environmental effects. And to solve this issue, what we propose is to have new observation from other isotopic dimension, what I call the isotopic anomalies. And for example, I'm going to use the clumped isotopes and the oxygen 17 anomalies. And how it's working, generally, we're going to build like a giant Sudoku. I'm going to have the information about isotopic composition of the coccolites for each dimension. And typically what I'm going to looking at is the correlation between this dimension. And I'm going to try to play with environmental parameters such as pH and metabolism in order to find what's the best combination of environmental parameter and biological parameter that resolve the system. So we wanted to test how these techniques can be applied to coccolites. And to do so, we elaborate a project in two parts. The first part was dedicated to culture experiments in a controlled environmental conditions. And this was done to verify the model. And the second part was more to see how this model can be applied in the past. And for that, we were looking at the last glacial, interglacial sedimentary record. Now I'm just going to focus on the first part for which the collaboration with the BIAM was essential. And so during the second year of my PhD, we elaborate together with Dan cultural protocol strategy. We had to adapt a bit what was used to be done because for isotopic measurement, I needed a lot of material. So we culture the coccolites in bigger volumes. And for that reason, we needed a large incubator. And finally, we cultivate 2 different coccolite species and 3 different strengths with different cell size at a fixed temperature. So during all the experiments, we measured pH, the cell, we follow the pH, cell concentration, and the formation of the coposphere during the experiment. So as you can see, just with the information of pH, we just from that, we can see that there's difference in metabolic mechanism between the different species. And during the experiment, we also sampled the water. And going back to the lab, I analyzed this water sample for estimated the growing environmental parameter as the delta frequency of the DIC and the delta 18 of the water. And from that, we calculate what should be the isotopic composition of the coccolites if their isotopic composition were only controlled by environmental parameter. Sorry. And this is what I represent here as the offsets from delta protein C composition or delta HMO composition. So these are really the first results. But what we have there is that we will see differences between isotopic composition registered in the coccolites plates as a function of the size of the coccolites. The bigger coccolites always enrich in delta 13C compared to the little ones. We also have an evolution of the delta 13C of the coccolites as a function of the concentration of the DAC and the pH of the media. And what we should do now is the quanti-isotope measurement. And for this, I'm going to use a mass spec, a bit modified, with a supplementary Faraday cup that allow me for measuring the mass 47. And I'm sorry, I don't have for the moment the results. So this is what should be done in the future. But it is also a really challenging measurement because in particular for coccolites, we should reduce the mass requirement for this analysis. So this is difficult. Sorry.
Thank you. We now continue with the presentation of Claire Rollion-Bard. She's collected. She can share her screen then.
Claire, tu peux partager ton écran, je pense.
Non, je ne peux pas le partager, je ne suis pas autorisée.
Ah.
Bon, sinon, ma présentation, je l'ai mise en ligne, s'il y a besoin. C'est bon. C'est bon, vous voyez bien?
Oui, parfait.
Okay. So today I will present some works that I did with Anna Yurikova from University of St. Andrews and Daniela Enkel from Leomar. And so we measure the carbon isotopic composition in culture brachiopod, and I will try to show you what we can learn from this isotopic composition. So why it is interesting to measure biocarbonate? So as you know, it is composed of calcium, carbon, and oxygen. And from that, you can learn a lot of things about the parameters of the ocean. So you can learn about the temperature, the oceanic current. You can have also some indication of the pH and the salinity. But the problem with this biocarbonate, as Marie presented, is that you can have some vital effect. So here it's an example for foraminifera. So it means the vital effect, it means that you have the modification of the record of environmental proxy by biological processes. So here it's an example for foraminifera. So it's made of calcite. And you can see that you have a huge difference in elemental composition between calcite which could precipitate from seawater and the foraminiferal calcite. So one of the big difference, for example, is the magnesium, which is really the most important in the inorganically precipitated calcite when it is not the case in foraminiferal calcite. So the problem that you have with the vital effect, so that you have a modification of the record of the proxies by biological processes. So it will lead to difficulties for paleoclimatological reconstruction. For example, we don't know what could be the vital effect for extinct species, and also is the vital effect constant through time? And if you have, for example, some sickness of the organisms or things like that. And also we have to determine what are the processes responsible for this vital effect. So for this, I studied the biomineralization processes via the geochemical signature. And I use in situ techniques like the ion microprobe and laser ablation. So here today, I will present only data from CCMs, so the ion microprobe. So here it's a picture of the ion microprobe. So it's a national facility which is located in Nancy. So you just have to write a project, and then you can have access at this machine. And so the interest is that you can measure directly on the sample without any chemical preparation and without any dissolution of the sample. So here it's an example of ion probe spot in a foraminifera. So I don't know if you see, you can see my my arrow.
Yes.
Yeah. So here you have the different spot of the ion probe, and so you can see that we can really measure a lot of things in one foraminifera. And here you have a SEM picture of one spot. So what is brachiopod and why we are interested in them? So brachiopods, they are constituted in two valves and they have a vertical symmetry. They are quite big and the average size is between 2 and 7 centimeters. But you can have extant species that are very, very big. And the interest of brachiopods is that you can find them in the geological records since the Cambrian. They are still present. In the present day, we have around 70 genera, but you can see here on this graph that they were really abundant in the past. And also their interest in that you can find them throughout the world and in the different oceans and at different depths. And brachiopods, for that, they are really often used in paleoreconstruction. And here you have an example of the strontium ratio reconstruction through time. And you can see that you have a beautiful reconstruction from them. So if you look at the shell of the brachiopod, it is constituted of two layers in calcite. The first one is the microgranular primary layer, so it's in contact with the seawater. And after that, you have the fibrous secondary layer. So for the moment, there is no defined motel growth for brachiopods. So here you have an example for bivalve, and in bivalve you can really see the presence of growth line, and like that you know exactly how it grows. But in brachiopods you don't have the presence of this growth line. So for the moment, the only growth models that you have for brachiopods is a mathematical model of growth, so which is this one. And here you have the addition of layer in the thickness, but also in this direction. So we wanted to test if this model of growth is valid or not. So for this, we use a culture sample of brachiopods that were cultured in BMR. Sorry. And there were some CO2 experiments. So you can see that they were first cultured at PCO2 of 2000 ppm. And after that, they were cultured, but in during less months at 4000 ppm. So the interest of this experiment is that in nature, the delta C13 of the DIC was around 0 per mil, but the culture, the delta C13 of the culture was around -24 per mil. So we have also some control experiments. So you can see that here you have the shell of the brachiopod, and here it is the parts that were grown during the natural condition. Then you have something like here that you can really sense on the shell, and after that you have the culture experiment, and here it is the shell for the CO2 experiment. Sorry. So what we can, what do we measure? So first in the contour experiment, so here you have the delta C13 and the number of points. You can see here the two profile. And you can see that we have the primary layer, which is really depleted in C13. It was something that was already measured. And after that, you have an increase until something like a plateau. And here you have the condition from the control experiment. So in the CO2 experiment shell, so here you have the shell, and here it is what we can see in the iron micropod mount. And you have here two little things that are when the brachiopod have the different control culture CO2. Sorry. So here. And so we measure in 3 different zones. So at the entire part, they are here just at when you have the change of culture, and here, which is normally the natural condition part. So in zone 1, so you have again the depletion in the primary layer, and then after that you have the increase. So we cannot measure a lot of things because you can see we don't have a lot of place here. And you can see the kind of values that we have, which reflect the DIC, the delta C13 of the DIC of the culture. In zone 2, so you have again the same. So the primary layer is really depleted in C13. Then you have an increase and something like a plateau. And in the zone 3, so it's normally in the part which grows only in the natural conditions. So you have here the delta C13 with the primary layer, always depleted in C13. And then you have the increase, the plateau, and then you have here a big decrease with value around minus 19 per mil. So it means that here you have the culture condition with this delta C13, which is really depleted. And we made some measurement in the other part of the valve to see if we can register the same things. And you have here again the same thing with primary layer. And then you have here this big decrease showing the culture condition.
1 minute.
Yeah, I saw. I try my best. So here it shows that we have an addition of a new layer on the parts that was already precipitated for natural condition, and then it validates the model of the mathematical model. Thanks.
Okay, now, Douville.
Bonjour à tous. Donc, après avoir parlé de coco, de brachiopod, on va parler de corail et donc des propriétés de calcification des coraux et principalement les effets d'acidification ou de réchauffement. Donc, on va essayer de mettre la présentation. Merci, Nan. Super. Impeccable. Merci. Donc, ces travaux sur les impacts des changements globaux et locaux se font dans le cadre de différentes missions à la mer qui ont eu lieu et notamment du projet Corrésilience qui vient d'être financé. On a différents types d'approches de géochimie marine, de la géochimie isotopique élémentaire mise en œuvre, de la datation des coraux via le carbone 14 uranium-thorium, et on étudie évidemment des effets de biologie et aussi de la modélisation pour étudier l'impact donc de l'acidification, des changements, mais aussi des effets locaux et éventuellement de la bioérosion et la notion aussi de résilience des coraux. Donc pour ça, on a bénéficié sur les dernières années de 2 grandes missions, une qui s'appelle Terra Pacifique, on a parlé de Terra Océan tout à l'heure, où on a pu bénéficier de collections et on fait des forages. Donc vous avez ici un exemple de colonie de corail, où dans cette colonie, on fore le corail pour récupérer des carottes de corail qui correspondent au squelette et qui nous permet de reconstruire dans le temps les propriétés de l'océan en termes de température, en termes de chimie des carbonates ou d'autres informations. Donc ici, on a eu Terra Pacifique et plus récemment, en 2019, on a eu une autre mission dans l'océan Indien autour des îles Éparses pour récupérer donc différentes carottes de corail donc qui correspondent à différentes colonies différentes. Donc, exemple de carottage, typiquement, on a travaillé sur deux espèces de corail massifs, les propriétés d'une part, qui sont classiquement utilisées en paléoclimatologie pour les périodes sur 200 ou 300 ans de reconstruction, et puis Diplostrea, qui est plus innovante, puisque c'est une espèce moins étudiée en paléoclimatologie, mais qui a l'avantage d'avoir un taux de croissance plus lent, et donc pour 80 cm, on peut avoir jusqu'à 200 ou 300 années de reconstruction, donc c'est intéressant d'utiliser cette espèce. Donc, la géochimie, donc on travaille essentiellement sur le squelette, donc en aragonite des coraux. Et c'est l'avantage de ces coraux, justement, c'est qu'on utilise d'abord la mesure isotopique et un élément qu'on n'a pas encore trop parlé, c'est l'isotope du bord qui nous donne des informations sur la géochimie de calcification et notamment sur le pH. Alors, le pH externe, celui de l'eau de mer, de l'océan, mais aussi le pH interne du fluide de calcification et donc qui va nous permettre d'avoir des informations, des indications sur les propriétés de calcification. Terrain favorable ou non favorable à la formation du squelette. Donc cet élément clé, il est essentiel, à la fois en termes isotopiques, mais aussi sur le forme bord sur calcium, qui va nous donner une deuxième information sur les ions carbonates. Donc on a le pH d'un côté, les ions carbonates, on peut reconstruire l'ensemble de la chimie des carbonates du fluide de calcification, et donc avoir des informations sur... Parallèlement à donc les mesures isotopiques qui se font sur le multicollecteur MCCPMS, on a des mesures élémentaires, donc pour le bord sur calcium pour les ions carbonates, mais aussi on a développé ces dernières un nouveau traceur de température dans l'océan, le lithium-magnésium, qui a l'avantage d'avoir une seule courbe de croissance pour dès lors qu'on n'utilise que des coraux sclératinières, entre moins 1 degré, parce qu'on a revisité récemment la courbe avec la thèse de Christian Cuny, jusqu'à 30 degrés, on a une seule courbe de calibration et on va dire une incertitude de température qui peut être entre 0,5 à 1 degré en régression de température dans l'océan. Donc, une fois qu'on fait ce travail de géochimie, c'est un exemple de série temporelle qu'on veut reproduire. Ici, c'est un exemple de 300 ans de reconstruction des paléo-pH de l'océan. Donc, reconstruit à travers des mesures isotopiques du bord le long d'une carotte de corail, ici prélevée en Nouvelle-Calédonie. Et on voit une chute du pH sur les dernières années associée donc, à l'acidification de l'océan. Le delta C13 a été ici mesuré simplement pour indiquer l'effet Sius, qui montre que c'est bien un effet anthropogénique qui est derrière cette chute du pH. À noter simplement qu'il y a 300 ans, on avait des pH qui étaient relativement faibles aussi. Et ce n'est pas la seule courbe qui le montre. Il y a eu 2-3 courbes comme ça similaires qui montrent qu'on n'a pas encore tout à fait bien compris la variabilité centenale des évolutions des propriétés des chimies des carbonates dans l'océan. Donc, donc les reconstructions dans le passé, on a besoin de travailler sur l'actuel. Et là, il y a un petit travail que je voulais mettre en avant à travers le travail de Marion Gehlen dans le cadre de sa thèse. Elle a publié un premier papier dans Scientific Report où on a récupéré toutes les carottes collectées Tara-Pacifique. On a travaillé sur le top core, donc sur les parties récentes, et on a voulu comparer les propriétés sur les parties récentes aux propriétés de l'océan local. On a d'abord vérifié que l'océan en représentait bien le milieu général de croissance des coraux prélevés. Et ensuite, donc, ce que je vous ai dit tout à l'heure, on travaille sur l'isotopie du bord pour retravailler sur le pH du fil de calcification et le bord sur calcium, les ions carbonates, qui nous permet de reconstruire l'ensemble de la chimie des carbonates. Donc, à travers ça, on peut étudier les processus de biomineralisation et quel facteur environnemental peut jouer sur ces processus de calcification. Donc ici, on a fait une stratégie de prélèvement sur 6 ans, on a comparé au milieu. Et pour information, les propriétés de croissance linéaire, ici vous avez des bandes de croissance sous les ans, et aussi la densité, puisqu'on s'intéresse à toutes les propriétés de croissance, tension, extension linéaire, densité et taux de calcification, sont reconstruites à travers 2 types d'analyses. J'ai voulu noter simplement l'accès à la plateforme d'Oseo juste à côté et aux listes où on essaye de faire de la microanalyse, c'est-à-dire de descendre aussi à l'échelle micrométrique. On a fait des travaux de quantification de la densité à 7 microns récemment. Et donc les résultats de cette analyse sur une quarantaine d'échantillons, 39 exactement, sur donc l'espèce Propriétés, c'est de montrer qu'à travers les différents paramètres environnementaux qu'on a étudiés, pH par exemple de l'eau de mer, salinité, température, sont les principaux curseurs qu'on avait regardés. On a vu le lien essentiel à travers les coraux Tara-Pacifique, c'était la température qui était le principal driver, là où le pH ne jouait pas, mais en fait on avait une gamme de variations du pH dans l'océan Pacifique faible et ne pouvait pas montrer un effet de gradient suffisant pour déceler l'effet pH. Inversement, on avait un gradient de 7 degrés de température, donc intéressant de voir l'effet de la température sur les propriétés donc du fluide interne de calcification en termes de chimie des carbonates et les propriétés de croissance des coraux ici présentées. Et ce qui a été ressorti, c'est qu'il y a bien un effet de température. Et ce qui était intéressant dans cette étude publiée, c'est que donc on avait 39 colonies de porythes, mais 6 colonies d'Hyplostrea, où sur le même site, on a également prélevé des porythes. Donc sur ces 6 sites, on a pu directement comparer la réponse à la température, et on a pu remarquer simplement qu'il y avait un effet notamment sur les paramètres oméga, ici, donc le degré de formation ou l'état de formation de l'aragonite des coraux, qu'avec la température, les propriétés du fluide de calcification étaient renforcées pour porythes. Inversement, on ne voyait pas ce renforcement sur l'espèce Diploastrea. Une autre façon de l'illustrer ici, c'est de montrer qu'avec la température ici, vous avez ici donc le pH, vous avez une chute dans les deux cas du pH interne de calcification, une augmentée des ions inorganiques dissous, et inversement, surtout des ions carbonates, vous avez une augmentation pour les porythes. De la même façon pour oméga, donc pour l'état de saturation, de formation de l'aragonite dans les coraux, et inversement pour Diploastrea, ce n'est pas le cas, comme si Diploastrea serait moins adapté à éventuellement, par exemple, le réchauffement climatique. De la même façon, on voit que porythes serait robuste et eux seraient une espèce capable, un genre de corail capable de s'adapter. De la même façon, une autre étude qu'on vient de sortir dans cet automne est d'étudier 2 colonies de porythes. Cette fois-ci, donc, on a prélevé les 2 carottes. Vous avez une première carotte, une deuxième carotte sur 2 sites. Un site contrôle ici et un site acide où il y a une différence, un delta pH de 0,18, ce qui correspond quand même à un peu plus que l'acidification actuelle sur les 100 dernières années. Donc, on a ici donc analysé la chimie du bord, les éléments de trace, on a reconstruit les propriétés de calcification et je vous fais simplement le résumé pour être rapide. On a observé simplement une chute régulière sur le site acide, sur cette carotte-là, des propriétés de calcification et notamment l'oméga, avec une chute de la densité directement associée à une chute des pH, du pH, enfin de l'ambiance pH plutôt faible dans cette région-là. Donc, plutôt sur le site acide. Et donc, ça, tout simplement pour dire, même si les coraux seraient adaptés éventuellement à un réchauffement, ils sont quand même impactés par l'augmentation du niveau de dioxyde de carbone et donc de la chute du pH dans l'eau. Simplement pour finir, on étudie également dans le cadre du projet Corps Résilience, donc qui a été financé par l'ANR, sur les effets locaux. Hein. On ne veut pas simplement s'assurer que sur la chimie des carbonates et la température, mais quels vont être les effets donc, de polluants, des métaux lourds, des nutriments ou aussi de microbes. Et on travaille aussi plus récemment sur les microplastiques. On travaille donc sur des coraux de terrain. On travaille en lien direct avec le CSM depuis des années pour des coraux de culture. On a mis pour la première fois en culture l'espèce Diploastrea, qui est une espèce massive. Ce consortium donc, est constitué du LSCE, du laboratoire de Villefranche, du Centre scientifique de Monaco, de l'Océan à Paris et d'EPOC, un laboratoire de paléoclimatologie à Bordeaux. Et donc, l'objectif principal de l'ANR, qui donc est en cours de développement, c'est d'étudier et de reconstruire à l'échelle du bassin. On essaye d'avoir une dizaine, une quinzaine, une vingtaine d'enregistrements, des variabilités en termes de pH, de la chimie des carbonates, de la température, mais aussi des métaux en termes de contamination de l'océan en Pacifique. Donc, une reconstruction, une modélisation aussi, et on voudra justement voir quelle a été la variabilité sur les 2 voire 5 derniers siècles. On a une carotte qui vient des États-Unis qui va recouvrir 5 siècles continus, et en fait, une meilleure compréhension de quel phénomène, quel facteur environnemental peut jouer directement sur les propriétés de calcification des coraux, ici les massifs, donc porythes et Diploastrea. Voilà, je vous remercie.
Merci. And now the last presentation, because Eric Peltier is cancelled his presentation. So,
Okay, then I talk for half an hour.
Allumer l'écran. Moyen moderne, des fois.
Good morning. So I'm Marion Gehlen. I'm a marine biochemist working at global scale, and it's a pleasure to present some broad aspects of that topic in this meeting. And it's great to see you all So I will show some examples on studies addressing the climate change impacts on marine primary production and biocalcification. So if I want to address climate change and impacts, one needs to upscale processes from lab experiments, as we have seen this morning, or here what I call the microbiologist view of the ocean ecosystem. This is a picture by Farooq Azam, which represents the microbial loop. It shows actually already some structure within the water, and the structure is set by the property of water, its viscosity in relation to the scale of the organisms, which are microbes. So it's a viscous environment where we have clumps, hotspots of activity around marine snow or detrital particles, etc. So this is a sketch which, as a biogeochemist, we are familiar with. It shows the vertical structure of the ocean, which obviously, of course, we all know is set by light penetration. We have the euphotic zone where photosynthesis takes place, driven by phytoplankton, raised by zooplankton, going up to fish, etc. So this microbial activity drives the biogeochemical wheel, the cycling of elements, carbon, nitrogen, phosphorus, silicon, and iron are shown here. And looking at the Earth system and feedbacks within the Earth system of these cycles to the radiative budget, so warming, we are looking on gas exchange with a particular focus of CO2, but also interested in oxygen. So how do we do this? Well, the ocean is part of an Earth system model. In this case, here we're using the IPSL coupled model. These are examples version 6, which contributed to the last IPCC report. The Earth system model represents the main components of the Earth system: the atmosphere, transport model, chemistry, the continental surface model ORCHIDEE, which was introduced yesterday, and there is a coupler between the atmosphere and the ocean component, which has physics, sea ice, and biogeochemistry. Zooming, So this is the biochemical view of the marine ecosystem, which is shown here in this sketch. So this is the Pisces model. It's also called Pisces in the Anglophone world. It is a model of intermediate complexity, which has 24 tracers, and it distinguishes 4 PFTs. So photosynthesis is driven or represented by small cells. Nanophytoplankton is a size class based. Diatoms, which are laser cells, which perform distinct biogeochemical functions in the ocean system. So photosynthesis here is limited by light and nutrients, so 5 nutrients. So our paradigm is, which is strongly hold in the community, that there is no limitation by CO2, neither present nor future, despite acknowledging that there is a different affinity between algae groups, but we don't represent this. So then we have grazing, we have mortality and export of detritus. So this is just an example of an outcome that we achieved with this type of model, actually a community effort, the coupled model intercomparison exercise, which then contributed to IPCC. So this is the emission scenarios that we use as a forcing for the Earth system models. The historical part, which is based on reconstructions, and then the projections. Here you see the evolution of atmospheric PCO2 in the called business as usual or the high emission scenario in pink, and here the green is a scenario that would correspond more to the Paris Agreement. And these reflect, of course, choices of our society, like societal and also strongly economical choices that we make. This is the response then now here of net primary production, ocean primary production as a function of time. Gray is the historical period, so corresponding to reconstruction, and here we have the projections. So this is the change in % of primary production relative to historical period for different scenarios. For the latest IPCC and the previous IPCC report, don't go into the details.
what we see is that for the high scenario, we have a decrease of NPP. But what we also see is we have a large spread across a multi-model ensemble of Earth system. So there is a really high uncertainty in the future evolution of NPP. So then we can look at what's behind, what could drive this change in NPP, and in general, how does the ocean react to climate change. In this panel here, you have various variables like SST, sea surface temperature, sea surface pH, oxygen in the intermediate layer, nutrients like nitrate here, and NPP. Again, as difference anomaly with respect to historical period. In a low emission and high emission scenario, the stipple shows areas where 80% of the models that have been analyzed agree in their response. So we see surface ocean fields, temperature and pH as a large agreement for properties that are set directly by the perturbation in the atmosphere. As we go deeper in the ocean for oxygen, we have some disagreements popping up here in areas where we have low oxygen levels and also oxygen minimum zones. So this reflects already the interplay between biogeochemistry and ocean circulation. And since all these models produce their own ocean circulation, which is different from one model to the other, they have their own ventilation processes. So they disagree in subsurface properties. Nitrate, it's again, we also see some agreement, but again, larger area where we disagree. So nitrate is just an example of a nutrient that then controls NPP, where we see a low agreement as also reflected in the plot before. But we can also notice, however, which is a persistent feature of this of these projections, is that we have a seesaw pattern with an increase in high latitudes of NPP and a decrease in lower latitudes in general. And this high increase here is attributed to an increase in growing season because of loss of sea ice in response of warming and an increase of stratification also due to warming. But these are well mixed zones. So if the stratification increases here, the algae will have more time in the sunlit ocean. So means the light limitation here is decreasing. And here the NPP decreases. It's also something, a kind of a robust feature, which as first order is also explained by an increase in stratification. But in this ocean area, this implies there is less nutrients that are upwelled to the ocean. So an increase in nutrient limitation as opposed to a decrease in light limitation, which at first order is not a full story, but explains the seesaw pattern. So take home messages from this. We have PFTs, plankton functional times. A PFT is described by a single set of parameters: growth, nutrient uptake, affinity, mortality, etc. But it's modulated by environmental conditions, by temperature, for instance. There's no plasticity in the response to changing climate. We have no representation of biodiversity and very little redundancy beyond PP and grazing. So which means that the functions might be unrealistically up or down regulated in these models. So now, how do we link the scales? This is an example of carbonate production. It shows how this is done. As we have here already, there is a relation to carbonate production to CO2, and it's the same also for photosynthesis. So what is key is the ratio of inorganic to organic carbonate production, which represent baseline value, which is modulated by light limitation. Because we don't have a PFT for carbonate production, it is assigned to nanophytoplankton, to light limitation, to nanophytoplankton growth, temperature control, a biomass criteria, which sets this PIC to POC ratio, which itself is modulated by the saturation state. And this function here has been fitted to a compilation of lab data. So that's the link between the large scale to actually the flask in the laboratory. Oops. Then in another study, this was expanded to assigning calcite production also to zooplankton that would then correspond to foraminifera and to aragonite in more soluble form, which are pteropods. It's the same basic philosophy underlying this then. And what we are looking at is the feedback to atmospheric CO2. And in this study, and this is actually also a very robust feature, and it can be proven with back of envelope computation, if you decrease calcification, the feedback on atmospheric PCO2 will be minor. This is exemplified here. We have a maximum feedback of, let's say, 12 ppm by 2100. Again, we use these models to explore. It's like a what-if scenario, to explore the space of possible outcomes. At this point, we don't have an explicit representation of calcifiers, but as I said, the feedback that I showed is robust across many, many studies. So there won't be a dramatic feedback. Doesn't save us. But what this study also shows that the feedback, the order of magnitude, depends on what type of calcifiers contribute to it. And so there is something, it hints at a potential effect of biotic interaction between different groups. And this is also very important. These models are biogeochemical models, so they don't allow to really probe or quantify ecosystem impacts. It's only about biogeochemical functions. But I think there are possibilities to work between the different communities here from BIAM and also LSCE or wider IPSL. And I hope I have shown the pathway of doing this, of upscaling from lab experiments to the global ocean. Thank you.
We have room for discussion now. Any questions or comments on the presentation of this morning? See a lot of possible interactions, as you said. No question?
I have a question.
Yes.
Coming to your work on the CCM mechanisms, it's quite intriguing. I mean, in our community, we know that it exists and that there are different affinities between allergens, but we don't expect this to happen. We don't actually expect the marine ocean, the marine NPP to be limited by CO2.
That's, I understand the point, and which is common in the literature, but the fact is that glycolate is present in the ocean, and glycolate, the major source of glycolate is photorespiration. Photorespiration is triggered by CO2 limitation. So what I think is that because we have to consider that CCM works, but it needs energy. And to some point, I mean, there's a hypothesis algae have to make the choice. I mean, between how do they use the energy? Do they use to concentrate? Do they use to fix? And what I guess is that under transient conditions, somehow the system is not able to react sufficiently rapidly to adapt to change in CO2, and that could explain that transiently we have a regime in which we trigger photorespiration. Because otherwise, how can you explain the glycolate concentration in the ocean? That's something, because that's kind of signature of photorespiration. There are not many, to my knowledge, there are no other source of glycolate than photorespiratory metabolism.
So it could be that would that then imply that the NPP, which is estimated from remote sensing, is overestimated? That if he
So what you wrote something about yeah, because you're talking about estimation based on chlorophyll fluorescence and the work of Falkowski and other.
yeah.
Yes, I think so.
That could be tested.
Because I think the rationale of Falkowski work was done on very limited conditions. On based this experiment, everybody agreed to use this correlation between fluorescence measurement and CO2 uptake. But, and it has been recognized by a few researchers that part of the electron flow, which is derived to, which is not used for CO2 fixation, but for oxygen consumption, have been ignored at the time. So, and I came to this because of this, when I saw this paper on glycolate, and I came to this paper on which I analyzed the glycolate concentration in the ocean. And despite the fact that bacteria, marine bacteria, proteobacteria have been described to use efficiently glycolate as substrate, still there is glycolate there. So which to me is, but I might be wrong. I mean, but I would like to discuss this point with.
I think we just perhaps at the coffee break, otherwise you.
Yeah, sure.
Yes, maybe we can continue during the, yes, Sylvia, you
So
want?
I have a comment.
Yes.
It's to everybody. Actually, in last yesterday and today, heard a lot of different talks. So my general question or comment actually is when we do most of work in BIAM, we work with pure culture in small scale, more in genetics and genome, protein level, regulation or cell level. What you do here in LSCE is more global level in the real field work. So I found that some how we can answer this question, I don't know the answer. So you can do a lot of models, but actually finally you cannot really test your model. And we can find a lot of fluxes, what we found in the lab, but we don't know in the real environment. Not only changes in temperature and light.
From the data are coming from pure cultures.
Yeah.
So, I mean, it's possible. I have seen also other studies done on other fluxes that I personally I'm not dealing with, like DMS and how the production of DMS, the genetic underpinnings of it. And then I think the British colleagues, they work really at the gene level, the isolated, I call it the genetic controls of the production of DMS and how that changes, how and you make the link between the production flux, the controls and temperature experiments. So I think it is possible, but it needs some thinking.
Yeah. See if we can build the bridges in this area to really the next step.
So I also like Eric, I also had studies ongoing with colleagues from Genoscope, Tara Ocean. But the challenge is to make the link between this omic data and the function.
Yeah.
Sure.
Because what we need in the model, we need it's a flux.
Yeah, exactly. Like go back to the glycolate story, we observed in the lab is secreted glycolate, and we looked in the literature in the ocean, it also secretes glycolate. But is it really for the same reason in the cell, in the pure culture? Is it what we don't know, the physiological reason of secretion in the real life, in the real field? Is it because to give some carbon sources to support the community growth? Or is it really to get rid of some, we consider a little bit toxic products to survive cells? So I think this we cannot yet to test that without co-culture and community work.
Thank you, Yongwa. Thank you, Tara. you can continue
Yeah.
while drinking coffee or. So thank you for the presentation, the discussion. And we have a break now and we start at 10, 11:10. Okay, in 20 minutes.
The 5 minutes later than-